Changing the chemistry is going to change the function. So, as we get in here, again, structure equals function is if you change the chemistry, you're going to change the shape of a molecule. If you change the shape of a molecule, you're going to change its ability to do the job it's supposed to do. So, let's focus on proteins because they kind of run the show. We have all these different structures of proteins, and they carry out all of these different functions. So when we think about these different structures, we have to get down to the amino acids because that's where the chemistry comes from. We have hydrophobic R groups, we've got hydrophilic R groups, we have ionized R groups, and it's these R groups that's going to alter the chemistry here. Again, going back to primary structure, we have the amino acids joining in a linear chain through covalent and peptide bonds. We get helixes and sheets through hydrogen bonding and secondary structure. And in tertiary structure, we're getting three-dimensional functional shape through those R group interactions. And again, as we look at these, if we look at each row here as a unique sequence of amino acids, they have a different R group in their primary structure, which would ultimately cause it to have a different tertiary structure. So each one has its unique chemistry because of the sequences of R groups and the order of the amino acids, and each one is going to end up being a unique shape. So when we have this sequence of amino acids, where do the sequence of amino acids come from? So if we work backwards, it would come from reading the triplet codons of messenger RNA. If we work backwards from there, the messenger RNA comes from the DNA and the genes. So when we look at these genes, each one is going to have a different sequence, which is going to create a different chemistry of amino acids and ultimately a different shape within the protein. If you change the shape of the protein, each of these enzymes are going to catalyze different chemical reactions because they will have different active sites. As we keep going here, what's unique and what's interesting here is that sometimes the same genes can even make different proteins. So these genes create this pre-mRNA. However, through alternate splicing, we can alter the structure and the chemistry of the messenger RNA, which will ultimately create a different three-dimensional shape within our proteins. And these two proteins, even though they can't come from the same gene, will have different functions. Now, most of the time when we start going back all the way to the DNA, problems arise during replication when we take one DNA and create two new strands. So the incoming nucleotides can get mismatched. And if DNA polymerase, it does sometimes make mistakes, but a lot of times it corrects it itself. But we can have these random mutations if DNA polymerase does not correct and attach the correct nucleotides into the DNA during replication. So if we have a normal DNA strand, we would get this sequence of amino acids. And we can have mutations. We have a point mutation because of a substitution of a single letter. We can also have frameshift mutations due to insertions and deletions. And one example of this is taking a look at hemoglobin. It has quaternary structure and it plays a very important role with the gas exchange of oxygen and carbon dioxide. However, if you have a simple point mutation here, all of a sudden you have sickle cell anemia because of the misshapen protein that comes all the way back to the chemistry. One last example here. We have phenylalanine turning into tyrosine. And this chemical reaction takes place because of this enzyme. And tyrosine turns into melanin because of this enzyme. So if we have a change in this enzyme, which makes it non-functional, what we'd end up with is the inability to do this chemical reaction, which would cause an increase in the amount of phenylalanine, which ultimately can become toxic to the nervous system, resulting in phenylketonuria. In this case, if we took out this enzyme, we would no longer be able to create this chemical reaction. So if we change the chemistry here, it makes it non-functional, which then would create albinism because of a lack of melanin. So if you change the chemistry, you will change the shape. If you change the shape, you're going to change the function.